This is the last part of the lecture for Unit 4. Biotechnology is the use of microbes to make practical products. Recombinant DNA technology is the intentional modification of an organism by natural or artificial means. There are a few goals of genetic manipulation. First is to eliminate undesirable phenotypic traits. Second is to combine the beneficial traits of two or more organisms. Third is to create organisms that synthesize products humans need. There are several tools used in recombinant DNA technology. Mutagens are used to induce changes in the genome and select for the cells with the characteristics considered beneficial. Reverse transcriptase will create a flow of genetic information in the opposite direction of conventional transcriptase, making RNA from DNA. And reverse transcriptase makes DNA from RNA. The RNA template is used to make cDNA or complementary DNA. It is easier to isolate the desired genes on the mRNA because there are more copies. In the eukaryote, the mRNA has introns removed, so it may be easily inserted into a prokaryote to make eukaryotic proteins like insulin. Synthetic nucleic acids can be used to produce DNA or RNA in a cell-free solution. This can be used to find the genetic code, to create genes for specific proteins, or to synthesize RNA or DNA probes to locate specific sequences. Probes are nucleic acids with specific sequences labeled so their location can be detected. Probes are widely used in medicine. Restriction enzymes are used to cut DNA molecules at locations with specific and palindromic nucleotide sequences. Once the DNA is cut, the other DNA sequences can be inserted into the places with the cuts. Vectors can be used to deliver a gene into a cell. Nucleic acids in viruses, transposons, and plasmids are all examples of vectors. There are several useful properties to vectors. They are small enough to manipulate in the lab. They can survive inside a cell. They also can contain a recognizable genetic mark and can ensure genetic expression by providing the required elements like promoters. When using vectors, a restriction enzyme cuts the DNA and vector into fragments with sticky ends. They are referred to as sticky ends because the ends will be complementary and readily stick together. Ligase anneals the fragments into a recombinant plasmid, and the plasmid is inserted into a bacterial cell. Finally, the bacteria is grown on a medium to select for what the cell has with the plasmid. PCR, or the polymerase chain reaction, is used to amplify DNA in vitro. It is a repetitive process that separates and replicates the DNA. The first phase is denaturation, and the DNA is heated to 94 degrees Celsius to separate the two strands of target DNA. The second phase is priming, a mix containing excess DNA primers complementary to the sequences near the end of the DNA polymerase, and an abundance of the four bases is added and cooled to 65 degrees Celsius. This allows double-stranded DNA to reform and provides the 3' hydroxyl group required for synthesis. In the extension phase, the temperature is raised to 72 degrees Celsius to increase the rate of DNA polymerase replicating each strand. The process is repeated over and over. After 30 cycles, needing only a few hours, over 1 billion identical copies are produced. While this used to be done by hand, it is now done automatically using a thermocycler. Here's a brief animation on PCR. One way to select recombinant cells is to use a probe to identify the clones. The probes will bind specifically and exclusively to the complementary nucleotide sequence and is identified by using radioactive or a fluorescent molecule as an attached label. In some cases, it is necessary to separate DNA. Gel electrophoresis separates molecules based on an electrical charge, size, and shape. DNA has a negative charge, so it will move towards the positive electrode through the agarose gel. The smaller the fragment, the faster it will move through the gel. The southern blot begins with gel electrophoresis. Next, it is transferred to a less delicate nitrocellulose membrane, and probes are used to localize the DNA sequence of interest, creating a fingerprint. The northern blot uses a similar technique to detect specific RNA sequences. So the names were actually derived from the first researcher who came up with the southern blot's last name was Southern. The northern blot was named not actually because of being northern, but because the other person's name was Southern. There are a few techniques for inserting DNA into cells. One is electroporation, which uses an electrical current to put a microscopic hole in the cell membrane, allowing the DNA in. Another one is protoplast fusion. Protoplasts are cells with the walls removed. When the protoplast encounters one another, the cytoplasms form a single cell with genomes of both cells. DNA can be injected into cells. This can be done with a gene gun powered by compressed gas or a blank .22 caliber cartridge. This can also be done using microinjection with a micropipette. There are many applications of recombinant DNA technology. Genome mapping is used to locate genes on nucleic acids. Restriction mapping will map the relative locations of genes. The DNA fragments resulting from cleavages by several restriction enzymes are compared to determine each fragment relative location to each other. After the fragments are cleaved, their sizes are determined using gel electrophoresis. Genomics is the sequence and analysis of nucleotides, basis of genomes. They used to be cleaved and mapped on a gel, but now cDNA is used, which is much faster. And here we've got a brief animation on restriction enzymes. DNA technology is also abundant in the medical field. Genes are inserted into microbes to synthesize proteins like insulin and interferon. Subunit vaccines are made by inserting antigenic polypeptides to make proteins to stimulate immunity without infecting the organism. The hepatitis B vaccine is an example. Southern blots may be used to screen for the presence of genes in genetic screening. DNA fingerprinting is used to identify individuals or organisms by their unique DNA sequence. This is widely used for paternity testing in forensics. Some techniques are still being researched, such as gene therapy to replace missing or defective genes with normal copies. With a xenotransplant, animal cells, tissues, or organs would be transferred into human tissue. There is the potential to inject a gene into an animal and have them grow human tissue or organs that would not be rejected. DNA technology has many uses in agriculture. It is used to produce transgenic organisms, which are recombinant plants or animals altered for a specific purpose. A gene from Salmonella conveying resistant to glyphosate or Roundup is used to create herbicide resistance. A gene for salt tolerance has been inserted to allow organisms to grow in salty soil. Excessive salt in soil decreases farmland available, and this organism is able to remove the salt to restore the soil. Freeze resistance has been created using a strain of Pseudomonas that has a polypeptide that prevents ice crystal formation. This is sprayed on crops. Bt toxin from Bacillus thuringiensis has been inserted in a variety of crops and kills insects. Bovine growth hormone in bacteria is given to cows to increase yield. A gene for beta-carotene has been inserted into rice to increase the nutritional value. The use of technology has raised several questions regarding ethics and safety. Is it ethical to raise animals solely to grow human tissue? The long-term effects of much genetic engineering are not yet known. Gene transfer may end up in organisms we don't want it to grow in, such as weeds. We may create new pathogens. The technology may be used for bioweapons. We may create proteins we don't know how to deal with. How should we handle genetic screening? Should genetic screening be routine or not? Who should pay for it? Who should know the results? Legislation is in the works to protect a patient from being denied insurance coverage and protect privacy with genetic screening. Another question that comes up is, should we be able to choose the sex of a child?